Welcome again to the course of structural biology. We have discussed about backgrounds, why we need to study structural biology, what about the architecture of protein, protein folding and all those things. Now we are actually going to where the Trojan horse is. We are going to talk about the techniques of structural biology. In this part, as I have already introduced myself to you as a protein crystallographer, we will discuss X-ray crystallography in detail. With that, I would also make comparative discussion of NMR and cryo-electron microscopy. Also, I will talk about other techniques like CD, like fluorescence, like small angle X-ray crystallography and others. But more importantly, I would also discuss how computational techniques would help solving or analyzing the data in all the techniques. So, welcome to the structural biology technique. Before going into the techniques, I just want to talk about the system, the body we have. If you look at, to understand biology, we have to understand at the organism level. From there, we go to the organ level. From there, we go to the tissue level. From there, we are going to the cell level. In the cell, there are macromolecules, DNA, RNA, proteins, and they are composed of small molecule. Also, there are small molecule metabolites, and ultimately everything comes from atoms and electrons. So, to understand biology, we should not be or could not be limited to knowing about the muscles, knowing about the bones. We have to go to actually in the atomic level, and it is very important because cellular function is an organized demonstration of millions of molecules. For normal functioning of the cell, it is extremely critical that these molecules communicate properly. So, communication is very important here. If you try to imagine this system as a factory where there are components, there are hierarchy, there are continuous synthesis, there are continuous destruction, you will feel something that here it is a factory, but there are no regulator. There are no one who is talking to stop or something. So, how biology communicate? How the systems in biology communicate with themselves, how they know when to start, when to stop. These are very important and these should be visualized in atomic level. And when I am talking about communication, because this is a introductory course, I do not know people like you who are taking how much they understand when I say communication. So, to provide you a proper example of communication, I have chosen to describe you a beautiful communication system in bacteria, and I would also show you how the communication is understood by us using high resolution structural biology techniques. When I say communication, when I say an urgent situation, especially in bacteria, I do not know again how much you could understand being the most powerful organism, most intellectual organism in the world. Let me take an example. Suppose you are drinking a glass of water and suddenly a drop of water falls in the table where you are sitting. Now, that is a real microscopic situation for you, not for the bacteria passing through that table where the drop actually falls down. For that bacteria, suddenly there is a sea of water created, and the bacteria was not even prepared for that. So, that is the problem of microscopic organisms, and because of that they have a total system which helps them surviving from emergency situations. One of them, which is very important, is called two-component system. A two-component system, as the name suggests, is made up of two proteins. One is a histidine kinase, another is a response regulator. A histidine kinase is a protein which actually stays on the or in between the surface of the organism. So, it have a part which exists in the periplasmic domain. It has another part which is in the transmembrane domain and a kinase domain in the cytosolic part. The periplasmic domain, it works as a receptor. There are different type of receptors responding to different emergency situation. When these receptor receive an external stimulus, it respond to that and convey the news to the bacteria and help bacteria expressing the proteins which could help them to survive. For example, if the external stimulus is sensing heat, it would communicate to the bacteria and bacteria will express heat shock proteins. So, how they communicate is beautiful and very interesting. Whenever this external stimulus bind to the protein histidine kinase, this histidine kinase dimerizes, and this dimerization help them to bind a ATP molecule. When the ATP molecule bind, they autophosphorylate in the histidine. And then I will talk about the further incidences, but let us take a look at the high resolution structure of these histidine kinases. You will see that they have the dimerization domain made by this orange color and green color alpha helical domains. These are the kinase domains. You will see mostly similar architecture of protein in all the histidine kinases. The transmembrane domain is not there, and this is also something, I could talk about the limitation of the technique, because their membrane part of the protein is not possible easily to solve through X-ray or other technique, because NMR, it is not solubilizing protein. So, it is only the cytosolic part which is solved, but this part helps you to understand the binding of ATP, the phosphorylation of histidine, and how this histidine is actually transfer the phosphate group to the response regulator. So, these are high resolution structures. So, coming to the next part, now the phosphorylated histidine transfer the phosphate to response regulator, which is having two domains, N-terminal domain and C-terminal domain connected by a flexible linker. So, the phosphate comes and bind to the aspartate residue of the N-terminal domain, and that leads to a change of conformation and opening up between the N-terminus and C-terminus. When this change of conformation happens, the C-terminus, which was initially bound to N, is now free, and being free, it interact with many macromolecules and small molecules. Like you see here, the C-terminus is binding to a DNA, here it is binding to a RNA, here it is binding to a protein, and here it is binding to small molecule. So, by binding to different biologically relevant molecule, it actually relay the signal the histidine kinase have received through its receptors. And not only that, if you take any of them like DNA binding, you see that the DNA binding is very symmetrical. If there is one residue here, after 10 base pair, you will get the same residue here binding to the exactly same residue of the DNA. So, there are different families. If you see NARL family, FOB family, NTRC family, ARA-C family, FIS family, they are all DNA binding, but if you closely take a look, you will see that the binding patterns are very different. Also, you would be amazed to know that they could also control the type of signal. So, depending on the binding affinity, depending on the way they are binding, they are actually relaying different signals. By that way, they actually provide a lot of different signals depending on the events happen. Just for a simple example, I talked about heat. It could be low heat, it could be like medium amount of heat, it could be very high amount of heat. Depending on them, they choose target, they change binding affinity, and in that way they do the relaying of the signal. And all these stories I am talking about could be understood in terms of solution of high resolution structure. So, it is essential in biology to look at atomic level if you want to look at the intricate mechanism of communication they are making. So, in biology, communication would be mostly performed through interaction. To understand the process of communication, we need to visualize molecule in the atomic level. That is the critical condition. We need to determine the proper arrangement of the atoms in those systems. But there are different level of interaction as well as size of macromolecules or their organizations are also quite different. What I mean is, if you think you want to look at one histidine kinase, and as I have shown you, you got it in the cytosolic portion. So, that have certain size depending on the molecule, they are like 300 to 500 amino acid, let us say. But now if you want to get the dimer, then it would be 1000 amino acid. If somehow you want to get the entire thing with the membrane, it would be a much larger assembly. Again, if you want to get together the histidine kinase along with the response regulator, they would be even bigger. If you want to get in the next level, the histidine kinase, the response regulator and the molecule it is binding, it even create bigger assemblies. Not to stop with that, sometime I talked just about dimer, they form multimers. So, in biology, most of the structural techniques we will be talking about, we are talking about atomic level, but at the same time larger assemblies like an entire virus. Today we are in the situation where COVID is a big problem. So, people are trying to get the structure, people are solving the structure of entire virus, and with that level of organization, with that level of different size difference, we need different techniques. So, based on those requirements, there are choices as well as advancement of techniques. I will talk about cryo-electron microscopy. Cryo-electron microscopy, when started, was actually in much lower resolution, but today if you look at, this is sensation. This is getting into atomic resolution. We will talk about that later. So, what should be the strategic plan to understand structural biology at atomic resolution? Biological system poses complexity. We have to identify the layers so that our understanding could be made at the fundamental level. Initially, the challenge was we want to understand the structures of the molecule, the simple molecules in like a single protein. Now, with the advancement of technique, with the dedication of great scientists, we have achieved a lot. We could now every day solve new structures. But understanding of biology is not being limited to that. Now we want more. We want from one protein to multiple assemblies, from multiple assemblies to the molecular machines, and so on. Studying the fundamental level would provide us high resolution pieces. Our job is to reconstruct them to build up the holistic picture so that we could understand not the function at the level of a single protein, but at the level of a machinery, like how a DNA polymerase is working with the whole replication complex or transcription complex, or what we got is the ribosome in its translational complex. These high resolution pieces would give us understanding of their mechanism of action at atomic level. Those understanding would be instrumental in understanding diseases. If we could understand how it is happening, we could also understand how the abnormality is coming. Let us say in a cancer disease, we understand which protein lose control, which protein is expressing without control, and how that is making abnormality and all these things. At the same time, we will know the structure of the protein of viruses or bacteria which we want to target as a drug development target. Also, now this is the arena of engineering. A lot of protein engineering, metabolic engineering, enzyme engineering is happening. Very recently, Francis Arnold got Nobel Prize for protein engineering. So, to do engineering, again we need to look at the atomic level. That is the necessary condition. So, techniques to solve structure of biological macromolecules, high resolution techniques, macromolecular crystallography, X-ray. As I told, I will talk about detail, nuclear magnetic resonance or NMR, cryo-electron microscopy or cryo-EM. So, let us take a brief comparison of them. If, as I started with, we look at the size starting from tissues to our atoms, it ranges from 1 millimeter to 0.1 nanometer. Light microscopy, which is a real fundamental tool for us to look at a lot of things, helps us to look at from the level of tissues to the level of organelles. But beyond that, it is not possible for light microscopy. We have cryo-EM, cryo-electron microscopy, which starts around 100 nanometer and currently goes to the angstrom level. Cryo-EM is a new generation sensation, and I will talk about that in more detail when I will talk about cryo-EM especially. X-ray crystallography, as I told, I want to talk about that in detail with the entire process. X-ray crystallography or macromolecular crystallography contribute mostly to the structure solution up to now. Nearly 90% of the solved structures are done by X-ray crystallography. It ranges from around 10 nanometer to angstrom level. Good structures are considered from 3.5 to 1 angstrom. NMR have relatively lower range. It cannot work with bigger molecule because it work with the tumbling, but NMR have its own application, especially when it consider dynamics, the dynamic property of macromolecule. NMR is really contributive, and beside protein, NMR have a universal usability, which also we will talk in details. Low resolution technique, there are many techniques. I will talk about circular dichroism, fluorescence spectroscopy, electron paramagnetic resonance, and small angle X-ray diffraction. But when I say low resolution, it seems these instruments or these studies are less efficient than high resolution. Not at all. High resolution structure solution techniques have their own application, but these techniques have another set of usability application towards getting solution level interactions. We will talk again. Today I will start X-ray crystallography. We will talk in details protein crystallography. It is a relatively young science, and together with nuclear magnetic resonance NMR and cryo-electron microscopy or cryo-EM, it is the main method for the elucidation of three-dimensional biological macromolecular structure like protein, like DNA protein complex, like virus. The use of crystals back to 1934, when Barnell and Crawford and Dorothy Hodgkin produced the first X-ray diffraction pattern of a protein, that of a crystalline pepsin, and Barnell and Crawford were awarded Nobel Prize. Their observation that pepsin crystal gave an X-ray diffraction pattern opened the subject of protein crystallography. Before people get to look at the proteins when it is in high concentration, some protein naturally make crystals. Crystals are good, crystals are easy to purify the protein, so they were used in that manner. But now it says that crystal also could have provide some meaningful information from its architecture. Although crystallography, when compared to NMR or cryo-EM, gives a more static description of the macromolecular structure, that is why crystallography is always criticized for, there are no limits in the size of biological macromolecules to be analyzed once they are crystallized. I will talk about this in details, but in crystallography, the main barrier is crystallization. A protein which is in solution would make a crystal or not. This makes X-ray crystallography the method of choice for studying macromolecules at an atomic level. You really get high resolution structure, you really get to see the molecules in real atomistic vision. In X-ray crystal structure, it analyzes the problem is to find out not only amplitude of the diffracted X-rays known as reflection, but also their phases. So, we need these two things, we need the amplitude as well as phase. Knowledge of both amplitude and phase allows to reconstitute the electron density of the crystal that give rise to the diffracted beam, which will convert it to model. The amplitudes can be deduced from the intensities of the diffracted X-ray, but the phases cannot be directly measured. So, you have a crystal, it is an array of molecules, you hit it with a X-ray, and when the X-ray hit the atoms, then you get intensity and you calculate amplitude. But if you think any ray or diffractive beam with wave properties, it could be in destructive phase or constructive phase. So, calculation of phase is very important, but it cannot be done through the normal X-ray crystallography experiment, and that is why it is called the famous phase problem. So, for protein crystal, this is a particularly difficult problem, and even measurement of X-ray intensities is not straightforward. So, in summary, protein crystallography is a method for studying the three-dimensional atomic structure of molecules. In this part of the lecture, our goal is to look at the milestone of protein crystallography advancement. It is a young science, but it have a lot of achievement. Around 30 plus Nobel laureates dedicated their life in doing research using X-ray crystallography or protein crystallography as a tool. So, new generation listeners, if you have higher ambition, this could be your technique. We will talk about process details required to perform protein crystallography. So, the process, how it is happening, and every techniques in structural biology is related to how to purify the macromolecule. In this part, I will talk about that. So, from how choosing a particular gene target to get a pure protein, which is equally applicable for NMR, cryo, or even with the low resolution structure solution techniques. We also talk about the application in the field of biological macromolecules, and we will compare, we already start comparing with NMR and cryo-electron microscopy. We will continue that comparison. We will talk about crystallography, we will talk about cryo and NMR, and then we will make a detailed comparison to understand what are the advantages and disadvantages of these three techniques. Coming to the steps in protein crystallography, as I told, to work with protein, it is always difficult because you cannot just start with protein. You have to go back and you have to start with DNA. So, your first work is to choose a proper protein target, but go back to the gene sequence, look at the gene sequence, then do some engineering. Nowadays, you optimize the protein so that you could, when you are doing the next step, cloning, you are going to get better product or something. So, if there are some portion which is not very much connected to the function, but not also part of the integrated structure, we try to chop them up. There are many strategies which I am going to discuss next. Then come to cloning, then overexpression, then purification. For structure solution techniques, purification is extremely critical, because you could only get good information if you have very, very pure protein in solution. Then coming to the topic, coming to the part I am talking about from the start to the end, which is crystallization, the biggest barrier in the success of protein crystallography. Mounting the crystal, how you take the crystal from the drop where you are going to set it to the diffractometer, the machine, how you collect data, what is your strategies, how to optimize data collection strategies. Then the phase problem I talked about, so how to work with that, how to determine the phase, what are the techniques, what are the problem, challenges, and how to solve it. Phase problem is the second biggest problem in crystallography. Then once you get the amplitude, get the phase, you will go for model building. Initial model building is not enough, so you go for further refinement through energy minimization and all. And once you have the refined model, you will check the quality, and ultimately you will do the data submission. Data submission, as I told, in protein data bank is essential, because once you submit the data, where you have to submit the model as well as the electron density, you will submit it, and that is not the end of the work. You will receive a validation certificate from them, and once you got a validation certificate from the PDB pupil, then only you are supposed to communicate the journal, which is the target of the entire work, to publish it so that the international community of scientists could have access to this structure. With this, I will finish today. If you have any queries, please write to us. More responses will coming from you. It would be helpful for us. And as I started with, I continue saying this, one of my inspiration to continue this course is to make a good generation of structural biologists from our next generation. Structural biology is becoming more and more critical towards biomedical engineering, drug designing, pharmaceutical, biotechnological problem and what not. So, more people would be getting interested, more people would be getting educated. We will be happy with that, we want that. Thank you.